So in the continuation of the regulation of gene expression, so in the last part we have studied about the lac operon. So lac operon when it turns on and when it turns off. So that is lac operon is an inducible operon where if the inducer is available then it becomes on, and if the inducer is unavailable then it becomes off. My advice or my suggestion to the students that they should go to the previous page and look to the schematical diagram of the lac operon in the presence of the inducer and in the absence of the inducer. Okay? So enzyme, so which enzyme, as you know that 3 structured genes are present in the lac operon: gene Z, gene Y and gene A. So different type genes or different type of structural genes that give the different enzymes. So the structural gene Z that give the beta galactosidase. So the enzyme beta galactosidase hydrolyze lactose. Beta galactosidase hydrolyze lactose. Into glucose and galactose. For obtaining energy. So this lactose is the substrate for this enzyme beta galactosidase. So, enzyme beta galactosidase that act upon lactose, that means that hydrolyze lactose. Lactose is a disaccharide that breaks the lactose into 2 monomeric unit by 2 monosaccharides, that is glucose and galactose. And glucose and galactose that give energy to the bacterial cell. So to obtain the glucose and lactose has to be hydrolyzed. So that means lactose, here the lactose is the substrate, and that's lactose, that is a substrate for enzyme beta galactosidase. which is the substrate here for beta-galactosidase, that lactose itself, that regulates the lac operon. That means that regulates the operon to be switched on or to be switched off. So that regulates switching on and off of the operon, because lactose here acts as inducer. If lactose induced, then the operon will become start or operon will become on. If lactose do not induce or lactose is unavailable, then the operon remains switch off. So here lactose actually acts as the inducer. So here it acting as the inducer. So lactose regulates the switching on and off of the operon. So now the question is whether lactose, as the lactose act as the inducer, then now the question is whether lactose is present in the medium or not. The medium in which the bacteria grows. So if the lactose is unavailable, then the operon system will remain switched on. If the lactose is available in the medium, then that lactose has to be transported inside the bacterial cell from the medium, from outside medium. So when lactose is available in the medium, then that lactose is transported into the bacterial cell by an enzyme permease. And permease is an enzyme that is produced by the Y gene, Y structural gene. So now the question is, unless lactose enters inside the bacterial cell, then the operon will not work. But to enter inside the bacterial cell, in order to enter, lactose should enter inside the bacterial cell. For that, the permease is required. But the permease will not be produced unless the operon will not work. Then what will happen in this case? Whether the operon will start automatically to produce the permease for allowing the lactose, or after entering the lactose, the operon will start. But if the permease is not available, then the lactose cannot enter inside the cell. Then what will happen here? This is a very important point. So, there is a very low level of expression So there is a very low expression, very low level of expression of lac operon. Because when lactose enter inside the bacterial cell, then the lac operon starts. But in order to enter inside the bacterial cell, permease is required. So permease has to be produced before entry of the lactose. When lactose enters, then the gene expression or the lac operon operate in a full-fledged. But before entry of the lactose, permease has to be produced. So and the permease only produced when there is expression, gene expression or lac operon expression. So there is a very low level of expression of lac operon only in order to produce the permease. So that is essential. For all the time, lac operon slowly operate only to produce the permease. So once permease is produced, permease allows the lactose to enter the bacterial cell, and once lactose enters the bacterial cell, lactose acts as inducer and it induces the lac operon to operate completely. So, but prior to that, a low level of expression of lac operon is essential for all time to produce permease. Only for production of permease, this low level of expression is required. So once permease is produced, then the lactose comes in, because permease allows the lactose inside the cell, and once lactose enters inside the bacterial cell, then lactose acts as inducer and make the operon switch on. So when the permease is produced, lactose will enter inside the cell. Unless the permease will not produce, the lactose cannot enter the bacterial cell. So, when lactose or allolactose, this lactose, either lactose or allolactose, both can act as inducer, either or. If not present inside the cell, that means if not present inside the medium or not present inside the cell, then the repressor protein is produced by IgG. Repressor protein always produced. It's not like this. If it is not present, it will produce. If it is present, it will not produce. It's not like this. The repressor protein always produced by the IgG. Already I told you that this IgG is the regulator gene. It doesn't have a switch. It doesn't have its own operator. It always, all the time, it produces the repressor protein. So the repressor protein, which is produced by the regulator gene, that binds to operator. So repressor protein is always produced. But whether it will bind or not bind, that will depend upon whether the lactose is present inside the cell or not present. If the lactose is not present inside the cell, then this repressor protein binds to operator. Otherwise, it cannot bind to operator because lactose acts as inducer, so it will bind the repressor protein. So when the lactose is not present inside the cell, then this repressor protein binds to operator because repressor is free now. If lactose comes in, then the lactose binds the repressor, so the repressor cannot bind to the operator. So if the lactose are not present inside the cell, then the repressor protein binds to operator, so operand becomes switch off. So if the operand becomes switch off, the RNA polymerase cannot transcribe the structural genes. That means preventing, when this operand becomes switch off, that will prevent the RNA polymerase from transcription or from transcribing the operand. So now, if lactose or allolactose, which acts as inducer, that will be available inside the cell. So in the presence of the inducer, repressor protein, here the repressor is free, because repressor binds to operator, because this lactose is not present here. Now the repressor is free, so if the free repressor can bind to operator, if the operator is bounded by the repressor, then the operator becomes off. But in the presence of inducer, the repressor is inactivated by the inducer. So I should say that you should follow the diagram in the previous page. In the absence of the inducer, what happens? In the presence of inducer, what happens? So in the presence of inducer, this inducer lactose binds to repressor, so the repressor becomes inactivated. So operator becomes switch on. Here, inducer was absent, so it cannot bind to repressor. So repressor binds to operator, so operator becomes switch on. But here, inducer binds to repressor, so repressor becomes inactivated. So the operator remains free. Operator is not bounded by the repressor, so the operator becomes switch on. So in this case, if the operator becomes switch on, that means the operand becomes switch on, lac operand becomes switch on. So regulation of gene expression begins. That means it allows the RNA polymerase, because RNA polymerase are present at the promoter site. So it should go to the structural gene. From promoter, it will move over the operator gene and go to structural gene for the transcription. So when the operator becomes switched on, that means operator is not bounded by repressor. Then in that case, the RNA polymerase which is present at the promoter site that move over operator and reach to the structural genes. So RNA polymerase move over operator and reach at the structural genes and the transcription begins. So these are actually the explanation of the diagram shown you in the previous page. So from this explanation, we come to know that lac operand is an inducible operand, because lac operand works or operates in the presence of an inducer. When the inducer induces the operand, then it becomes switch on. That's why lac operand is called as inducible operand. lac operon is an inducible operon. Lac operon is an inducible operon because it becomes switch on and off depend upon the inducer. That's why it is called as inducible operon. So inducible operon, why it is called as the inducible operon? Because it becomes switch on in the presence of inducer. So when inducer comes in, then it will induce the operon. So the operon becomes switch on and the transcription begins. That's why it is called as inducible operon. And also it acts in the catabolic pathway. Because lactose is the substrate, lactose is inducer as well as the substrate. So lactose breaks down into monomeric units glucose and galactose. So lactose has to be catabolized. That means this operon is for this operon when works, the lactose breaking enzymes are produced. So lac operon occurs in a catabolic pathway because lactose here breaks down or catabolized. So that is catabolic pathway and also known as negative regulation. Why it is called as the negative regulation? Because the lac operon stops when the glucose and galactose is accumulated inside the cell. So continuously lactose breaks down, glucose and galactose monosaccharide units are produced, glucose and galactose are utilized in the respiratory pathway to produce the energy. So when it becomes the beyond the bacterial cell, beyond the power of the bacterial cell, the glucose and galactose, if not further utilized, then it will be accumulated inside the cell. When these will be accumulated inside the cell, then this operant system will stop. That means this operant system acts in a negative regulation. So with this we come to the end of the lac operon, which is an inducible operating system or inducible operon. So in the bacterial cell, other operant systems are there. Another operating system is the repressible operating system or repressible operon. The example is the TRP operon, tryptophan operon. So here you know that in lac operon, the operon remains in switch off condition. When the inducer comes, then it will make the operon on. But in the repressible operon, in TRP operon, it will always on the on. It is always the TRP operon remains on always, but on always, and it will produce the tryptophan amino acid by a series of reaction. So it will produce many enzymes that act in a series manner. Ultimately, the end product is tryptophan. Tryptophan is the amino acid which is utilized in the bacterial cell. But when the tryptophan is accumulated in much amount, that tryptophan will stop the operon. That means that in the repressible operon, the operon remains switched on always to produce the tryptophan, the end product. When the end product is accumulated in much amount or excess amount, that tryptophan itself will stop the operon or it makes it switch off. So in lac operon, it is from switch off to on condition. In TRP or repressible operon, it is from switch on to off condition. When the end product accumulated, that will make it switch off. That means the end product acts as a repressor. That's why it is called as repressible operating system or repressible operon. So the repressible operant system, it is found in anabolic pathway. This anabolic pathway is the constructive pathway. We have studied in the lac operant, it is the catabolic pathway because here lactose is degraded, but here the tryptophan is synthesized. So it acts in a constructive pathway, that is called as the anabolic pathway. And this repressible operant, the example is the TRP operant. The repressible operant system includes tryptophan, or shortly it is called TRP operant, inside the E. coli bacteria. And in this operant, TRP operant, many enzymes are synthesized which help in the synthesis of tryptophan amino acids. That means if this operant works, the tryptophan amino acid is synthesized. So it operates in a anabolic pathway. This operant is normally switched on, but when the end product means tryptophan is accumulated, then it will make the operant switch off. So here, the TRP operant, or it is a repressive operating system, this is normally switched on, and when it becomes switched on, the enzymes are produced, and the enzymes goes on synthesizing the tryptophan amino acid. Because when this is switched on, normally it is switched on, always it is switched on, unless the end product is not accumulated. So when it is switched on and the tryptophan production continues, and tryptophan is being used inside the bacterial cell, but due to increased formation or due to increased availability, either it is formed inside the bacterial cell, or if it is supplied from the outside source. If we provide the bacterial cell tryptophan, then availability will become more. So either increased formation or due to availability of the tryptophan or the end product, that tryptophan acts as co-repression, because tryptophan is going to stop the operant. So tryptophan, which is amino acid, it acts as co-repressor, which activate aporepression. which activate aporepressor. And operand becomes switch off. So, in the lac operon, we have studied that regulator gene produces a active repressor. But here in the TRP operon, regulator gene produces an inactive repressor that is known as aporepressor. So, when the tryptophan is accumulated, the tryptophan is the end product. When it is accumulated inside the cell in much quantity, then tryptophan acts as co-repressor. And co-repressor binds with the aporepressor, so the repressor becomes inactive. And the repressor goes and bound to the operator gene, so that the operand becomes switch off. Otherwise, the operand always on to produce the tryptophan. If you look at the schematic representation of the TRP operon, is a segment of DNA which acts as the operating system. They are the structural genes. So 5 structural genes are present. A, B, C, D, E. You can write it also TRP. TRP A, TRP B, TRP C, TRP D and TRP E. And this one is the operator gene. This is the promoter gene. This is the regulator gene. And this is the promoter for regulator. Promoter of regulators. So this mark here, this regulator gene has no operator. So, the regulator gene produces an mRNA, that is TRP mRNA, where here the promoter having the RNA polymerase that will move over the regulator gene. So, the regulator gene transcription begins. When the transcription begins, the TRP of mRNA is synthesized. From from TRP mRNA, a repressor is produced that is called as aporepressor. So this one is the inactive repressor. So this aporepressor cannot bind to operator gene. Here is also the promoter. Here is another RNA polymerase present. This RNA polymerase move over the operator and go to the structural genes to produce the enzymes that will move over this. So TRP E that will produce enzyme E. Enzyme will conduct certain reaction. TRP D produce enzyme B. TRP C produce enzyme C. TRP B produce enzyme B. TRP A produce enzyme A. So this will occur in a series manner. The various enzymes, 5 types of enzyme that control the 5 steps in the chain reaction. Ultimately, the end product is produced, that is tryptophan. Because look here, this regulator gene are not producing active repressor. If it produces active repressor, then this repressor will bind to operator and the whole process stops. But this regulator gene produces the inactive repressor, that is aporepressor. So aporepressor cannot bind to operator. That's why the operator becomes on or remain on, so that RNA polymers move over it and the many enzymes are produced, and ultimately the end product tryptophan is produced. So when this tryptophan becomes excess after utilization inside the cell, tryptophan is being continuously produced in the bacterial cell and tryptophan is utilized inside the bacterial cell. After utilization, if it will remain or it will become excess, then this tryptophan acts as co-repressor. And this aporepressor and co-repressor will be combined. So this will, the co-repressor, it will activate the aporepressor, so that this aporepressor become active repressor. And this active repressor will bind to the operator. So this active repressor will bind to operator, so this TRP operator becomes switch off. So when it will become switch off? When this aporepressor become active repressor. And how it will become active repressor? When the co-repressor combine with the aporepressor, then the aporepressor turns on, or repressor becomes active repressor. Then only active repressor can bind to operator and the whole system becomes switch off. Otherwise it remains on the switch on condition and continuously formation of the tryptophan. When tryptophan is accumulated or end product is accumulated, it will make the operating system switch off by binding to aporepressor. So this is the TRP, a tryptophan. This whole we can call it as a repressible TRP opera. So in earlier we have studied lac operon, that is inducible lac operon. Here it is repressible TRP operon. In inducible lac operon, that remains switch off and it becomes on by an inducer. But in the TRP operon, it remains switch on condition, but by accumulation of the N product itself act as a repressor, so it turns the operand switch off. So in inducible operand, that is from off to on, in repressive operand, that is from on to off. So this is the over of the regulation of gene expression.